There is a fungus that Tibetan herders have been chewing for 15 centuries before crossing mountain passes where the air is too thin to breathe. In 1993, Chinese athletes consumed this same fungus and shattered 3 world records in 6 days. Western media screamed doping. The International Olympic Committee demanded answers. The coach finally revealed their secret. Not a laboratory stimulant, not a designer drug, a mushroom that grows on dead caterpillars in the Himalayas. Scientists tested it. The results were undeniable. This fungus alters human metabolism in ways that seemed impossible. Then the world wanted it. What once cost pocket change now sells for more than gold by weight. The people who discovered it can no longer afford it. This is about what happens when ancient knowledge meets modern science, when performance enhancement comes from the soil instead of a syringe, and when the rarest medicine on earth becomes too expensive for the people who discovered it. September 1993. The National Games of China in Beijing. The women's distance running events are about to rewrite the history of human performance. September 8th. Wang Junxia runs the 10,000 meters. She crosses the finish line at 29 minutes and 31.78 seconds. The previous world record, held by Norway's Ingrid Christiansen since 1986, falls by an astonishing 42 seconds. This is not incremental improvement. This is a performance leap that defies every model of athletic progression. September 11th, 3 days later. Wang Junxia runs the 1,500 meters. She finishes in 3 minutes and 50.46 seconds. Another world record. Shattered. September 13th. Wang Junxia again. The 3,000 meters. She runs 8 minutes and 6.11 seconds. The previous record falls by 16 seconds. 3 world records in 6 days. All from the same training group. All from athletes who had been competitive but not dominant just months earlier. The international athletics community erupted. The accusations were immediate and harsh. East German coaches had systemically doped female athletes in the 1970s and 80s, destroying lives with steroids disguised as vitamins. The Chinese team's sudden dominance triggered every alarm. The International Association of Athletics Federations demanded tests. The International Olympic Committee sent officials. Blood was drawn. Urine samples collected. Everything analyzed under the most sophisticated anti-doping protocols available in 1993. The results? Negative for anabolic steroids. Negative for erythropoietin. Negative for stimulants. Negative for every banned substance on the list. Completely clean. But the accusations did not stop. Some Western coaches insisted the Chinese had developed undetectable designer drugs. Others claimed the testing protocols were insufficient. The conspiracy theories multiplied because the alternative seemed impossible. How could athletes improve this dramatically without pharmaceutical intervention? Coach M.A. Junren held a press conference. The international media expected denials, deflections, maybe accusations of Western bias. Instead, M.A. revealed his training methods in surprising detail. The athletes trained at altitude in Qinghai province, where oxygen levels are significantly lower than sea level. This was not unusual. Many distance runners use altitude training to boost red blood cell production. But M.A. described something else. A strict diet that included turtle blood soup, caterpillar fungus tea, and a traditional Chinese medicine called Cordyceps sinensis. He claimed these natural substances, combined with brutal training regimens, explained the records. The Western press dismissed it. Turtle blood and fungus? This sounded like mysticism, not sports science. In 1993, Western sports medicine focused on synthetic supplements and pharmaceutical-grade interventions. The idea that a mushroom could enhance performance seemed absurd, but M.A. Junren was not offering mysticism. He was describing a fungus that had been documented in Chinese medical texts since the 15th century, and he was about to introduce the world to one of the strangest organisms on Earth. Cordyceps sinensis does not grow like normal mushrooms. It does not sprout from rotting logs or decomposing leaves. It grows from death, specifically from the mummified corpses of ghost moth caterpillars. Here is how it happens. In the high alpine meadows of the Tibetan Plateau, at elevations between 10,000 and 16,000 feet, ghost moth caterpillars burrow into the soil during late summer. They are preparing to overwinter underground. A cordyceps spore lands on the caterpillar. The spore germinates. Fungal filaments called hyphae penetrate the caterpillar's exoskeleton and begin to spread through its body. The fungus consumes the caterpillar from the inside, slowly replacing its tissues with fungal mass while keeping the creature alive. By winter, the caterpillar is dead, but it remains upright in the soil, its body now a fungal mummy. When spring arrives, a dark brown stalk erupts from the caterpillar's head and pushes through the soil toward sunlight. This stalk, called the fruiting body, releases spores to infect the next generation of caterpillars. The entire organism, caterpillar corpse and fungal stalk together, is what traditional medicine calls Cordyceps sinensis. In Tibetan, it is known as Yartsa gunbu, meaning summer grass, winter worm. For at least 6 centuries, Tibetan herders have been harvesting this fungus. They dig through meadows in late spring, searching for the tiny brown stalks barely visible above ground. Once found, they carefully extract the entire specimen, caterpillar and fungus together, and dry it for preservation. Traditional Tibetan medicine prescribed cordyceps for fatigue, weakness, and respiratory issues. Herders chewed it before long treks across high mountain passes. Elderly patients consumed it to restore vitality. It was expensive even then, but available to those who knew where to look. Chinese medical texts from the Qing dynasty describe cordyceps as a tonic that tonifies the lungs and kidneys, stops bleeding, and resolves phlegm. By the 18th century, it had become a luxury item in imperial courts, reserved for emperors and elite families. But the mechanism was unknown. How could a fungus that grows on dead caterpillars enhance human performance? For centuries, the answer was simply tradition. It worked because it had always worked. Then scientists began testing it. The first modern research on cordyceps appeared in Chinese medical journals in the 1980s. Researchers extracted compounds from the fungus and tested them on laboratory animals. The results were unexpected. Mice given cordyceps extracts showed increased swimming endurance. Their time to exhaustion extended significantly compared to control groups. This was not a placebo effect. Something in the fungus was altering physiology. Further studies identified the active compounds. Cordyceps contains nucleosides, particularly adenosine and cordycepin. These molecules are structurally similar to adenosine triphosphate, ATP, the primary energy currency of cells. Here is how it works. Mitochondria are the cellular powerhouses that produce ATP. Cordyceps compounds enhance these energy factories through 3 simultaneous pathways. First, they increase ATP production by improving mitochondrial efficiency. Second, they enhance oxygen utilization, allowing muscles to extract more energy from each breath. Third, they improve blood flow by relaxing smooth muscle and blood vessels. Some studies also indicate anti-inflammatory properties and immune system modulation, which helps athletes recover faster and avoid overtraining. The measurable results are significant. A 2004 study published in the Journal of Alternative and Complementary Medicine tested cordyceps supplementation in elderly Chinese subjects. After 12 weeks, subjects showed a 7% increase in maximum oxygen uptake and improved metabolic thresholds. These are measurable, significant changes in aerobic capacity. Another study in 2010, published in the International Journal of Medicinal Mushrooms, found that cordyceps supplementation increased time to exhaustion in endurance exercise by approximately 9% compared to placebo groups. These are not the 30% increases claimed in exaggerated headlines, but 7 to 9% improvement in endurance is enormous in competitive athletics. Olympic medals are decided by fractions of a percent. When combined with elite training, optimal nutrition, and altitude adaptation, even small metabolic advantages compound into record-breaking performances. The 1993 Chinese runners were not just eating cordyceps, they were training at altitude for months. They were following brutal workout schedules that Western coaches later described as inhumane. They were consuming carefully controlled diets. Cordyceps was one factor among many, but it was a factor, and that was enough to change everything. After 1993, demand for cordyceps exploded, not just in China, but globally. Athletes, health enthusiasts, and aging populations all wanted access to the fungus that might enhance energy and vitality. The problem is that Cordyceps sinensis cannot be cultivated. The fungus requires a specific host, the ghost moth caterpillar, which only exists in high-altitude Tibetan ecosystems. The environmental conditions are precise. Temperature ranges, soil composition, altitude, and seasonal timing all matter. Scientists have successfully cultivated Cordyceps mycelium in laboratory settings. This produces a fungal biomass that contains some of the same compounds found in wild cordyceps. But it is not the same organism. The laboratory-grown version lacks the full spectrum of compounds present in the wild fungus caterpillar complex. Wild cordyceps can only be harvested by hand. Collectors spend weeks in remote alpine meadows, crawling through grass, searching for tiny brown stalks. One person might find only a few dozen specimens per day. The work is grueling, the yield is low, and the harvest season is short. The price trajectory tells the story. In the early 1990s, wild cordyceps sold for approximately $12 per pound in rural Tibetan markets. By 2000, $100 per pound. By 2010, $10,000 per kilogram. Today, premium wild cordyceps sells for $20,000 to $50,000 per kilogram. More expensive than gold by weight, more valuable than platinum, one of the most expensive biological substances on Earth. The price explosion has devastated Tibetan ecosystems and communities. What was once a supplementary income for herders has become the primary economic driver across the Tibetan plateau. Entire villages now depend on cordyceps harvesting. During harvest season, schools close. Families migrate to high meadows and set up temporary camps. Children as young as 6 join the search, crawling through grass for hours each day. The work is back-breaking. The competition is fierce. Overharvesting has become severe. Studies show cordyceps populations declining in heavily harvested areas. Meadows that once yielded hundreds of specimens now produce dozens. The fungus reproduces slowly, and intense harvesting disrupts its life cycle. Conflicts have erupted over access to productive harvesting grounds. In some regions, violent disputes between villages have resulted in deaths. The Tibetan Plateau, once a place of pastoral calm, has become a gold rush landscape where desperation drives behavior. And the people who have harvested cordyceps for centuries, who understand its traditional uses, who know the meadows and the rhythms of the fungus, can no longer afford to consume it themselves. The global market has priced them out of their own medicine. The supplement industry responded by cultivating Cordyceps mycelium in controlled environments. This laboratory-grown Cordyceps is affordable, widely available, and marketed as equivalent to wild specimens. But research suggests significant differences. Wild Cordyceps contains compounds that laboratory cultivation does not replicate. The caterpillar component contributes unique proteins and nutrients. The high-altitude environment affects the fungus's chemical composition. Laboratory Cordyceps may provide some benefits, but it is not identical to the wild organism. Studies comparing wild and cultivated Cordyceps show variations in adenosine content, polysaccharide profiles, and bioactive compounds. Some research indicates wild Cordyceps has stronger effects on ATP production and oxygen utilization. Other studies find minimal difference. The truth is likely somewhere in the middle. Cultivated Cordyceps provides measurable benefits. It is a legitimate supplement with real physiological effects, but it is not the same as the fungus that Tibetan herders harvested and that Chinese athletes consumed in 1993. The wild organism, shaped by millions of years of evolution in extreme environments, contains a complexity that laboratory cultivation cannot yet fully reproduce. So what do we take from this story? The 1993 records were not just athletic achievements. They were the moment the West was forced to acknowledge a metabolic truth the Himalayas had held for 15 centuries. Tibetan herders understood Cordyceps' enhanced endurance without knowing the biochemistry. They did not need ATP charts or mitochondrial diagrams. The knowledge was empirical, passed down through generations, validated by experience across hundreds of years. The tragedy is not that we learned its secrets. The tragedy is that in learning them, we destroyed the ecosystems where it grows and transformed a communal medicine into a luxury commodity. Cordyceps is not a miracle. It is not a performance-enhancing drug. It is a fungus that produces marginal metabolic gains that compound into championship performances when combined with elite training. The fungus still grows in the Himalayas. The herders still harvest it. The meadows still bloom each spring, but the medicine of the people has become a trophy for the elite. The athletes who broke records in 1993 proved that performance comes from more than laboratories and designer drugs. Sometimes it comes from the soil, from organisms forged in the harshest environments on Earth, from knowledge so old we forgot it was wisdom. If this story opened something for you, subscribe and hit the bell. Every like and every share helps preserve this knowledge.